welcome to my YouTube channel Science Academy. Today I will tell you about the DNA. First of all, the main question is, what is DNA? Where is DNA found? DNA is in every cell of every living thing. It is found within the chromosome of the cell. Chromosomes work to build protein and assist in duplication or division of the cell. Basically, DNA is present in every cell, in plant, in animal, in every cell. Its function is changed in plant and in animals. In human body, the somatic cell and reproductive cell, two cells are reproduced by mitosis and meiosis. In somatic cell, there is double helical structure is present, and in reproductive cells, in egg and sperm, there is single helical structure is present. It is present in the chromosome in the nucleus. Its main function is to build protein and assist in duplication or division of the cell. The unique structure of DNA allows it to be the hereditary molecule and allows it to store instruction for directing cell activities. The DNA stores the hereditary molecule. For example, it restores the parental hereditary material, and so the children are unlike the parent. In plants, it also restores its hereditary molecules. That's why the baby plant is like its parent plant. Then I tell you about the abbreviation of DNA. DNA is made up of deoxyribonucleic acid, made up of sugar, which is deoxyribose. One OH is removed and hydrogen is replaced, and macromolecule store the genetic material. DNA stands for deoxyribonucleic acid. It is a long molecule made up of monomers called nucleotide. The monomers unite together to form the long strand of DNA. One nucleotide is made up of one phosphate, one deoxyribose sugar, and one base. What is base? Base is adenine, guanine, thymine, and cytosine. When it is in term of base, it is known as thymidine, cytidine, guanosine, and adenine. Phosphate, when there is one phosphate molecule, it is known as AMP. When two molecule, it is ADP. When three molecule, it is known as ATP. Adenine triphosphate, adenine diphosphate, adenine monophosphate. Now this is one nucleotide is united with another nucleotide, which is guanine, thymine, adenine. The only difference is of base. It may be adenine, guanine, thymine, and cytosine. The twisted ladder shape is known as double helix. This double helical structure, which is made up of nucleotide base, sugar, and phosphate, is twisted around each other to form the double helical structure. Now what is nucleotide? Nucleotide, the backbone of DNA, is formed by alternating sugar and phosphate held together by a strong bond. This sugar and phosphate are joined together formed by a strong bond, which is known as phosphodiester bond. It is very strong bond and it is one nucleotide. The rungs of the ladder are formed by the four nitrogen bases and are held together by weak hydrogen bond. The bond between the nitrogen base, for example in adenine and thymine, is double bond, which is weak bond which can break easily. In cytosine and guanine, there is triple bond, which is also hydrogen bond. It is also a weak bond. The nitrogen bases are joined together by hydrogen bonds, and the bond between sugar, deoxyribose sugar, and phosphate is known as phosphodiester bond, which is very strong bond. Now the last question, how does DNA work? The four letters of DNA made up codon. These chemicals are repeated in the various orders over and over. These codons made up genes. These genes tell cells how to make a protein that control everything in the cell. Codon is the makeup of three nucleotide bases, for example ATC, GTA, ACG, etc. These are the code of a specific gene which is used to make protein. This protein performs a specific function in our body. That's why in DNA, information are stored in the form of codon. The answer is, if the code in DNA is TACGATTGA, it is replicated in RNA form, and in its opposite form, T is opposite of A, thymine is opposite of adenine, adenine is opposite of thymine, cytosine is opposite of guanine, guanine is opposite of cytosine, etc. The gene in DNA is replicated in RNA form in its opposite form. When this DNA information is replicated on RNA strand, it is converted in its opposite form. When this RNA is again used to form DNA, its opposite is formed, which is same as of DNA. In this way, the genetic material is stored in code form and used to store the information of DNA. In this way, a new cell is formed which is same as the parent cell. In this way, my lecture is complete. If you like my video, then subscribe my channel. Thank you.